the last few decades, metabolomics has really become a lot more popular and a lot more prevalent, so you see it a lot more. It's used in a lot more publications, and a lot more people are doing it, including non-experts and people who just sort of do metabolomics on the side. And while metabolomics has become a lot more prevalent, it hasn't exactly reached the status of something like genomics or proteomics, which has really found clinical applications and is really um, there's a lot of standardization there. And metabolomics, everybody kind of seems to be doing their own thing. Metabolomics has a lot of reproducibility issues, and there have been some efforts to fix this, but those have also not been great. And the end result so far is that we have an omics technique with a lot of potential that's sort of just kind of weird, and it's often not used in a way that's really the best. And I think that the issues that cause these things are very understandable. And I think that if people knew what they were, then they would be able to maneuver them a lot better and understand their own data a lot better. So I want to discuss these with the title Why Most Polar Metabolomics is Not Great. And I want to go through each of these issues and discuss them with you. So first things first. So when I say that most of the polar metabolomics is not great, this might sound very negative, but there have been quite a few recent publications that really highlight the issues. For instance, there's a publication that says that at least 85% of the proposed biomarkers are simply statistical noise. So these are serum metabolomic studies that deal with cancer, different types of cancer, and they went through all of these metabolomic studies and they checked, okay, the biomarkers that are being suggested for these cancers, are the other studies finding them as well? And so in 85% of the case, 85% of the studies, this wasn't the case. So these biomarkers are unlikely to actually be biomarkers, and they're just random noise. Now, another paper that came out this year said that greater than 70% of all peaks found in metabolomics are likely in source fragmentation. So these aren't actual peaks. This is just noise. And so these two things kind of tie. They kind of. It's the same. It's basically the same problem. So a lot of the peaks are nonsense, and these peaks are then suggested as biomarkers, which are then nonsense. They're useless. Now, you can debate these percentages, but I just want to show this to show you that it isn't 10%. It isn't 5% of things that aren't great in this field. It really, truly is the majority of the studies out there that aren't good. So first I want to compare metabolomics to other omics, and I'll focus on MS-based omics. So I won't focus on genomics or transcriptomics, but I really want to focus on the MS omics. So first, metabolomics really is a different beast than these other two main omics, so lipidomics and proteomics. So when you deal with lipidomics, lipids tend to form these clusters. So you'll have a lipid class, for instance, TGs or PCs, and they'll have these fatty acid tails, and these fatty acid tails will just increase by usually 2 carbons. And so you'll get these very predictable clusters of lipids that you can use for your annotation. So you won't just identify one individual PC lipid, but you'll identify the entire cluster, and you can do retention time corrections on these things because they're very predictable. You tend to use things like C18 columns for the LC, and the mass range is usually suitable for MSMS or mobility. Now, with proteomics, all the analytes that you're analyzing, so lipidomics still has quite a few classes, right? So you have the things like TGs, which aren't really, they're neutral, and then you have things like PCs and PEs that do have a charge. So you have some chemical diversity in this sense, but still they are fairly similar. Now then you have proteomics, and all the analytes there are peptides of similar size. So this is why we use trypsin. So you get all these analytes that are really quite similar, and they're also highly predictable. So you just have the human genome. You can just predict whatever peptides are produced using the genome and using your knowledge of what trypsin does. And then the chromatography is very straightforward. So you tend to use just a C18 column, and all of the peptides, they just form this mountain of peptides typically. And then you use multiple peptides to identify a single protein. So it's not like you need a single specific peptide and that's it. You piece these together, or the software pieces these together to identify a full protein. So you have a sort of check there as well. So straightforward chromatography into a combined thing that ends up being an ID. Now, metabolomics really is different from these two things in the sense that metabolomics is really, it's individual analytes that all have very little connection to each other. So you'll have something like lactate, but also something like AMP, for instance. And there's no real logical connection between them. They're chemically very different. They're in a large mass range, and they tend to be very small. And so when something is already really small, like an alanine, which is only 3 carbons and a few oxygens. Is it 3 carbons? Yeah, I think it's 3 carbons. Then fragmenting it doesn't really work. And if you turn on ion mobility, you tend to just lose all of your peaks because it just isn't very compatible with this. So, which I already kind of highlighted. So you have a large diversity of chemicals. So you have NADH here, you have amino acids, you have a sugar with a phosphate on it, nicotinic acid, ATP. These things are all really different. Glutathione, they're their own thing. So there's not there's no cluster of glutathione. Amino acids, I mean, it's a class, but they're all chemically really diverse. That's kind of their point. So each one of these things you really have to identify and analyze as its own thing. And so metabolomics has a lot of these, has a lot of issues, and I want to go through them sort of one by one. So first, because you have this very large chemical diversity, it's very difficult to optimize your LC method for this. So if you optimize something for lactate, then you're unlikely to be optimizing it for ATP as well. And so this means that doing an LC separation in itself is just very difficult. There have been some really good columns that came out that tend to measure all of these compounds quite well. So for instance, the BEHG Hillick column, which is by Waters, and then the Merck Zixi Hillick column. So this is the Waters column, and this is the Merck column. So as you can see, there's the silica thing, and then there's a linker, so it has a bit of a tail, and then there's a Zwitter ion. So you have a permanent positive charge and a permanent negative charge. Here the same thing, but the order is reversed. So if you look at a C18 column, it's just a C18 tail, right? So the interactions that this can have with metabolites that come along, they're really very limited. So it's just stick in the tail or not. But with something like this, you have quite a long tail. So this is just physically, there is a barrier. And then you have this thing with 2 charges on it, but it's 2 opposite charges. So something that's positively charged, does it really want to interact with the column? Well, kind of, because there's also a negative charge, but also not really, because there's a positive charge. So you get sort of a weird combination of effects that's controlling the retention time within these columns. Additionally, because these columns this is much more complex than a C18 column, so a C18 column was just a rather inert carbon tail, these things are more, well, they appear to be more sensitive to things like damage and contamination. And as I said, because of the chemical diversity of all of your analytes, it's very difficult to optimize your method for each one of them. So you'll have ugly peaks. This is just a fact of metabolomics. Like, I really do not believe that anyone has optimized their metabolomics method in a way where you have really nice peaks. Also, these columns tend to have very broad peaks, just as a feature. Additionally, the columns have quite unpredictable retention time shifts, because there isn't just a single variable that's controlling retention time, but it's this thing between sticking in the tails and also the charges. It's difficult to predict retention time shifts. So for instance, what I see often is that the nucleotides with their phosphate groups, these are already beginning to shift, but for instance, the lactate at the beginning of the spectrum is just perfectly fine, or the lysine at the end of the spectrum is still also just perfectly fine. So it's specific classes that start to shift and start to move around or start to lose their nice peak shape, but it's not all of them. Additionally, I've noticed that especially the Merck columns that we have, And I'm sorry, but we've had just in the last 4 years, we've had a lot of supply issues, and also the quality control just isn't great. So we can run 400 samples and then they're done, and within these 400 samples, you see this massive shift in retention time. So just in general, chromatography in polar metabolomics isn't great. It's worse than in lipidomics or in proteomics as a rule. Now, I want to show you some automated integrations. And whenever you show these kinds of things, it's like I said, it's always easy to say, oh, you should optimize this, or oh, just do this or do that. But I just want to really show you the type of chromatograms that are at the basis of polar metabolomics. So this is something that the automated integration by Bruker task integrated. So it says that this is a adipic acid, which is of course great. This is a lovely peak. And for instance, here at the bottom you have the mass for alpha-ketoglutarate, and you can see that it's attempting to integrate a peak, but as you can see, this isn't really a peak. It's just sort of, well, enthusiastic noise, I would call it. And so you see a couple of these things. So here it took the peak at 6 minutes, here it took the peak at 6 something, and here it's even further. You might say, well, it should have taken, well, maybe this peak, but then, so that's, here there is a peak, but here there isn't. And this is all from the same project, run right after each other. So this isn't like months of work. This is within the same run. And again, you can tell me, oh yeah, but we do retention time corrections and all of these kinds of things. But just knowing and seeing that this is the type of thing that's underneath this, and this also isn't necessarily fixed with a blank subtraction, because this tends not to be in the blank, but it is in the samples. So just knowing that these kinds of peaks are underneath what's being integrated, especially when you use automated integration programs where you never even see the extracted ion chromatograms, which I really think that the majority of people don't look at their chromatograms. They just look at whatever the peak areas are that they get from automated programs. I think it's important to show just how bad some of these things are, and basically this is not that you cannot integrate this. I would throw this out of if I was doing this the analysis of this set, I would throw this out instantly. So you don't have alpha-ketoglutarate. I'm sorry. So this is another example. So you have glucose. So sugars are always difficult. You have these specialized columns for sugars, but they don't really work very well for the other metabolites maybe. So well, you just try to make the best of it. So glucose on all of the columns that I've tried for polar metabolomics, it has just been ugly. So I'm assuming that that's just the case. If anyone out there has lovely peaks for glucose and everything else, please let me know. But I sincerely doubt that, we all have kind of the same things at our disposal, right? We have acetonitrile, we have water, and we have ammonium bicarbonate or ammonium formate or acetate or whatever. And that's basically it. So I'm assuming that we're all dealing with kind of the same ugly peaks. So this is the internal standard that I have. So I have the internal standard for glucose, so 13C labeled. And this is the peak shape, so I know that this is the peak, this ugly thing. Now the automated software, it made this out of it. So here it indeed integrated the whole thing, then it took the left side, then it took the right side. And you also see that the peak isn't really stable from sample to sample. So that's not great. Now, so this is just that was the first point. So chromatography in general isn't great. Like I've shown you now our worst one, so I don't want you to think that all of them look like this. Some are really pretty, but they're just a non-insignificant part of your data set is just going to have really ugly peaks. Now, the second point is that many important metabolites have close isomers. So I just put a few up here, but the sugars are, of course, very well known. So you have the glucose, mannose, galactose, fructose. They all have the exact same mass, citrate, isocitrate, so it's TCA cycle, and then itaconate, citriconate, etc., etc. And then the various amino hydroxybutyric acid type things, which there are a ton of those. So a lot of the metabolites that you really care about have very close isomers. And what this means, so what this means is, of course, that you have multiple peaks, or you expect, if you can detect them all, that there are either multiple peaks or that you have not separated them, and that these peaks are overlapping within your chromatography. So the third one, and I think this is one of the biggest ones, is in-source fragments. So as I said in the in this thing that came out this year, more than 70% of all peaks observed are likely in-source fragments. So if you're just running some sort of database across all of the peaks that you've measured, I mean, so the majority of those things are just nonsense. Now, already in 2015, I saw this paper which showed this comparison. So they had 130 metabolites, so 80 of them were fairly straightforward, and then they have 29 that were affected by structural isomers and 21 that were affected by in-source fragmentation. So this is a lower number than this, but I think it's actually maybe worse than they saw if I look at my own data. And this is something you would typically see. So an in-source fragment of ATP, for instance, will still have the retention time of ATP because the fragmentation happens after the chromatography. And so if you can separate the ATP and ADP in your analytical run, ours looks fairly similar to this actually, then you would see this peak show up in the ADP spectrum. So in-source fragments really are a big problem, and I kind of want to go into a few examples just to show you. So they're a problem because despite diversity, so I said that there was a lot of chemical diversity, there's a lot of overlap. So chemically diverse, if you put a phosphate on something, then it's chemically diverse in the sense of chromatography and behavior in the cell, etc. But it still shares like the majority of its structure with each other. There's just a phosphate on it. And phosphates tend to break off really easily, for instance. So despite the diversity, these shared structures are there, so in-source fragments from one thing end up in the next. And I kind of also want to highlight that with lipidomics, if you have in-source fragments, typically a fatty acid tail will fall off, for instance, so you can go to a lyso species. But in general, if they both fall off, in general, you sort of they go out of the cluster, so you don't really. They might be less of a problem. I'm not saying they aren't a problem, because I can already hear our bioinformaticians be like, well, actually, but it's not the same as here. So, and with proteomics, for instance, you have fixed peptides that you know the kind of peptides that you're expecting, and any fragment of that will be unlikely to be a peptide that you would expect to see, for instance, or it's just too small to even be detected, etc. But now we're dealing with the small stuff. Like, we are looking actively at the small stuff, and as things fragment into smaller pieces, you'll just logically see them more here. Now, this is one of the examples. So arginine to ornithine. So you see this part of the structure they just completely share. So if arginine fragments, then you just have ornithine. So if you look to the chromatogram, you'll see ornithine here, and this is the arginine in-source fragment. So this broke off. It's still at the retention time of arginine and broke off. This is a random peak. I have no idea what this is. I always see this. I don't think it's a metabolite. I think this is also an in-source fragment of something. And a very important point here that I really, really want you to remember. So if you think that all of this was a bit of a ramble and you think, oh, I don't really care about any of this, please remember this. So this in-source fragmentation happens before the MSMS fragmentation. So if you are using MSMS to be confident about your peaks, which is something that I often get from reviewers, they ask like, oh, do MSMS to be confident. And I just really think it doesn't work for polar metabolomics. And the reason is this, because if I fragment this, so if this fragments in my source to this, I can then very successfully run an MSMS experiment on this and just conclude that I've indeed found ornithine. I will just say that it's ornithine. I won't say that it's arginine. And so MSMS really, really doesn't help you against in-source fragments, unless you already have the retention times. But if you already have the retention time difference, then you also don't need the MSMS stuff. So I think MSMS is very unhelpful here. And I kind of want to give some more examples just to show you how big the problem is within polar metabolomics. So these are all metabolites that kind of fragment into each other. So we have ADP ribose here. So there's a ribose and an ADP. So ribose goes into ribose. It also goes into ADP. ATP, of course, fragments into ATP. This is NAD, and NAD, if it breaks off here between the phosphates, which happens a lot, then you get NMN and you get AMP, which is this one. So ADP also fragments into AMP, of course. They all fragment into adenosine, which then fragments into adenine. Then you have ribose, which can come off of this. It can also come off of the adenosine, of course. Then you have ribose diphosphate, which can break up into ribose phosphate. And you can have PRPP, which can also then break up into ribose diphosphate or ribose. Well, you get the point. Like, all of these things share structures. And when you hear the number that I just said of 70% in-source fragmentation, which again, we can debate the number, but it's a very big number, then this kind of makes sense, right? So all of these things share structures, so they're all fragmenting into each other, and yeah, it's not great for peak identification. So a few more examples just to show you the issue, because you might already have some ideas like, yeah, but I know this, I can do that with my bioinformatics, and I'll figure it out. But I want to show you how complex some of these are. So this is malate. So this is a lovely peak, malate. This is fine. Like for Hillock, this is really a fine peak. And then you have fumarate, but actually this isn't fumarate. This is actually a malate in-source fragment. So it's if you take off H2O, then you get fumarate. So okay, cool. So malate fragments into fumarate. So what is really fumarate? Well, it's actually this tiny little peak here. So I've spiked this with a standard, and this is actually fumarate. So first things first. So MSMS doesn't help. Again, it would just fragment this mostly, and we would just say, yeah, I've identified fumarate successfully, confidence score of 3 or whatever they use. Lovely. I've done a great job. But it just isn't. It's just malate in-source fragment. And the automated software also consistently just picks the wrong thing because there's such a massive peak next to a tiny one, and he's like, don't you care about this one? And no, I don't, but it will just continue to do this. However, so you can still, like, let's say you have a brilliant program and you can get it to just do this one. Maybe you set the retention time window really small because you see, oh, it's very large. How dare you have such a large retention time window? You should put it way smaller. Well, again, I just want to point out chromatography is horrible, so your peaks are shifting. Your peaks aren't shifting very predictably, so it's also difficult to do retention time correction. Maybe there's some fancy stuff out there, but it's a difficult thing that you really have to actively do. But also, so the chromatography is bad. So if you look, this is actually one of the spectra. So we have this beautiful malate in-source fragment right here, and we have our fumarate right over there. So that's lovely. I can integrate this, right? I can just manually, oh, wait, where's it going? And it's gone. So this doesn't work. So how this would show up if you do automated integrations and you don't really check them, you might be able to integrate this, and then you would see like a small peak area, and then suddenly you would see like an explosion of a peak right here. And again, you could say, oh, maybe with the smoothing, and maybe, like this is already problematic in terms of quantitation, but this is just pointless. Like this just doesn't make any sense. So these kinds of examples, I hope, illustrate how difficult this is to isolate and how difficult it is to fix. Okay, so the next one, I see cis-aconitate or trans-aconitate a lot in datasets from other groups. I don't know how they are measuring this because citric acid tends to just fragment into aconitic acid, and I think so. This is just the peak from citric acid, and then there's a lot of other ones, but they don't really match up with the standards that I've measured. So I think they're in-source fragments from something else or something. But this entire spectrum is basically just nonsense. I've never integrated this peak. And if somebody out there is doing it and they're like, yeah, no, definitely it is this, I would love to hear it, but I just have my doubts. So then a lovely, I think this is the last example, but this one is really, really lovely. So you have itaconic acid, which is kind of a high P metabolite as well now, because it regulates things in the immune system. So it does things with macrophages and stuff. So it's a really nice metabolite to have. So I spiked a sample. So this is just a normal sample, and then I spiked this sample with the standard for itaconic acid. Well, lovely. So here's the peak. So that should probably be the peak. So then I measure another sample, and it looks like this. So is this itaconic acid? Maybe. I don't know. Is it this? And is this the shoulder? Is that the... I don't know. But then, like I said earlier, we also have mesoconic acid. So I checked, and actually this peak matches up with mesoconic acid. So I did another spike, and this peak went up. So I now know the left is itaconic acid, the right is mesoconic acid. Lovely, beautiful, well done. But actually, no, because this peak is completely dominated by 2-hydroxyglutarate in-source fragmentation. And so this looked like mesoconic acid, but it isn't. It's predominantly 2-hydroxyglutarate. And actually, I can measure the standard, but within samples, I'll just never see it because it's always covered by this thing. I actually figured this one out because I was looking at a heat map of a project that we did, and the mesoconic acid peak and the 2-hydroxyglutarate peak, they moved exactly the same throughout all of the samples. So I saw that they just perfectly correlated with each other, and I was like, wait a minute. And again, there's an H2O loss between the two, so this is nonsense. But I see these in a lot of other people's metabolomics datasets, and I know how messy this spectrum is. So I really doubt that people are actually integrating this properly. Now, another point that I really want to make, which is kind of separate to the issues themselves, but a lot of people do not use internal standards. I still see this all over when I go to conferences. You can typically see it if they show you a graph and the values that they show in the box plots are really high. So if they are just basically raw MS values, so if it says like a count of like 1 million or something, then you tend to know like, you didn't use internal standards, you're just using raw values. But there's a lot of reasons to use internal standards, and in fact, I would argue that not using internal standards in metabolomics, polar metabolomics, it basically means that your data is almost useless. Because first of all, like the easy stuff. So there's no correction for sample errors if you don't use internal standards. So if you are pipetting and you're pipetting less of one sample than of the other, if you've added internal standards beforehand, so labeled versions, so with carbon-13 or with the nitrogen label beforehand, then if you pipette a little less, well, you also pipette less of the internal standard. So if at the end you divide every peak area by an internal standard, then you correct for this loss of sample. Now, and this is a big one. So if you don't use internal standards, there's no correction for ion suppression. And this is a major one because you're looking at different groups typically. And the intensities of peaks aren't just determined by how much is in there. They're determined by the environment that the peak is in at the time of measurement. So if something comes off at like 7 minutes at the column, and there's also just massive other peaks there, then the peak itself will be suppressed. And this is really a severe, like, this isn't like, oh, you'll lose 5%. Like, this can really just, you can just lose peaks. And if you're not using internal standards that match the retention times, then what you can get is that in one sample group, there can be something in there at a certain retention time that's just suppressing all of the other ions at this retention time. Sorry. So if you don't use internal standards, you'll just be looking at the ion suppression. So you'll just see, glutamate went down in this group of samples. But actually, glutamate didn't go down. Actually, it just stayed the same, or it might have even gone up. But something else might have gone up even more and suppressed your metabolite. For instance, if you add a compound, if you give a cell culture a compound or whatever, and it ends up in very high concentrations in your cells, which can happen for some compounds, this can just suppress certain metabolites, and you'll be like, oh yeah, adding this compound depletes this metabolite. No, it just interferes with the analysis of this metabolite. If you use a correct internal standard, this will be at the same retention time. It will also be suppressed, and you can correct for this. And you can also check the internal standard to see, hey, wait a minute, all the internal standards on this at this point are way lower, so there must be something going on analytically. So that's a problem. The other problem is that you also have no reliable retention time reference. As I showed you, some of the chromatography can be wild for some peaks. Having internal standards means that you have some reference, like, oh, wait a minute, the retention time of the internal standard has also shifted half a minute. So you have some road signs where you can orient yourself. And this one is a bit like but having internal standards just means that you have more reliable QC options. So you can using internal standards, you can check your retention times. You can check sort of the stability of your internal standards, but as I just said, they're supposed to be suppressed as well, so that's but you can use them to check at least if your analytical run went fairly well. So internal standards are highly important, as there are almost no clusters and patterns that can help aid identification of peaks. So I really feel like you need correct internal standards. So what are good internal standards? Well, you can't have internal standards for every compound in metabolomics. That's just crazy. It just won't work. It's just too expensive probably too. But you need representatives of each class that you're interested in. So have a few nucleotides, have a few amino acids, have a few sugars, a few sugar phosphates, some organic acids, some like the smaller stuff, etc. And this is the most important part. It should be spread out across your entire analytical run. So I've seen a lot of studies as well where they're like, yeah, we used internal standards, but they'll use one. So they'll use, for instance, labeled glutamate for the entire thing. And they'll have a run of 21 minutes, and glutamate is usually quite a nice peak, let's say at 8 minutes or something. If you're correcting something at the retention time of 1 minute for an internal standard at 8 minutes, you're doing the opposite of fixing your data. So you are controlling indeed for the pipetting stuff, but if there's a lot of suppression around 8 minutes because a big bump came off there, but nothing like that came off at 1 minute, and now you're suddenly correcting this for the thing at 8 minutes, then you're really introducing all kinds of noise in your data that you really don't want. Like, this is actively messing up your data. So please don't do this. You need to have internal standards in at least roughly the whole retention time area. So we have around 30, I think, across a 15-minute run, and they're nicely spread out and they represent classes. So we indeed have these kinds of things. Now, also check, you don't want any interferences with endogenous metabolites, so always measure, always have one sample that's just a blank, add internal standards, run it through the entire sample prep, and at the end, just analyze it as you do everything else, and then always check if you're interested in a certain peak, if it's not also in this blank with the internal standard. We also found out, for instance, that that's not internal standard related, but we found out that, for instance, succinate is in one of the cleaning products that we use in the dishwasher. So everything that touched any glassware that went in the dishwasher now contained a whole bunch of succinate, which was very interesting. And so check that there's no endogenous metabolites. Also check the fragments. So some of the peaks I showed probably are also fragments from internal standards. Now, the last point I want to make is that automated checks tend to fail. So things that other omics might use, they don't work for metabolomics. So retention times and peak shapes on Hillock, they just have poor reproducibility. Hillock is complex, etc., etc. MS doesn't really help. Like I said, you have these in-source fragments, and even if you can fragment something, like it doesn't necessarily tell you that much. A lot of these fragments are just really generic, and they're just not very interesting. I've gone through a list by others who've done MSMS validation, and they're like, oh, now I did MSMS, and I'll show you later. It just doesn't work, and it just doesn't really help you. So we just don't do MSMS. We do MS1 now, and I peak pick things by hand because, yeah, it just doesn't add anything except some, well, yeah, no, it doesn't add anything except complexity of your method. And they're too small for ion mobility. So if you ever want to have a blank sample, you don't have a blank sample, just turn on the ion mobility and you'll see all of the intensities basically disappear for the small stuff. I'm not saying it can't be fixed. I'm not saying there aren't machines out there who are doing this properly, but our experience and the experience of some of our collaborators has been that if you turn on ion mobility, you just lose all the small stuff. Now, because of all these issues, so we've gone now through all of these issues, and because of these issues, you kind of get these a lot of the data sets that I've seen from other groups, they're sort of all the same thing. So they all have like things in common that I've noticed, patterns. And I want to discuss these, and I want to go through some of them. So one of the things that I see is that I see a lot of duplicates from isomers. So I'll see people annotate all of the sugars, for instance. Okay, cool, but I tend not to see peaks for all of the sugars, or they are indeed in-source fragments from, for instance, sugar phosphates, where the phosphate falls off and they're now in the sugar spectrum. I also tend to see things like trans and cis aconitate. So as I showed you, there were multiple peaks there. They just probably annotate a random peak there. Beautiful. And some studies I've even seen, like I had a PI at my desk once, and he was like, you gave me a metabolomics set, and it had only 120 peaks in it, and now I found this article, and they have 5,000 metabolites. Like, basically asking me, why are you so crap? And I was like, oh, why am I so crap? So I went through the article, and it turned out that they just annotated, like any mass that they found, they would just annotate every possible isomer of this. So if they found one sugar peak, they just took the same peak, and they just annotated it with every possibility. Then you get up to 5,000 metabolites, but it doesn't mean anything. And again, most of it is in-source fragmentation. So then you get these semi-duplicates. So you have duplicates from isomers, then you get semi-duplicates from the in-source fragmentation. So the fumarate-malate problem. So they'll have fumarate and malate, but really one is just an in-source fragment of the other. There's also a lot of integration of what I call the garbage heap of metabolomics. And these are just these small generic molecules that don't really have any sort of like... They don't have any personality, really. So it's like a benzene ring with a little acidy thing on it, or like all of the hydroxybutyric acidy type things. And these are extremely contaminated with the in-source fragmentation stuff. So any amino acid that breaks down tends to end up in this sort of thing. And they're metabolically also not very interesting. In niche cases they are, but in general it's just not great. So this is just like the small crap. Just don't do this. Then there's a lot of drugs and exogenous metabolites, so salicylic acid, malic acid, which isn't endogenous as far as I'm aware. It's also a fumarate isomer, so you just get another fumarate. That's lovely. And things like xylitol. Whenever I see these databases, whenever I see these data sets from others, there's so many artificial sugars in there. Like they're in all of the data sets, they're in all of the standard boxes that you can buy. So there's always like xylitol, erythritol, and whatever. And then I want to say, so a lot of it is like a lot of the stuff that is there isn't great, but then some of the stuff that really should be there is absent. So, for instance, in this reproducibility crisis paper, there was this, even glutamine, which was the most commonly reported and abundant amino acid, was only reported 59 times across 244 studies. Now, this is mind-blowing to me because I've looked at hundreds of metabolomics data sets over the past few years, and I think I can count on one hand when I could not detect glutamine. Like this is always in a data set. So this is just in all cell types, in every, like only in like the weirdest matrices have I maybe not found glutamine, but like maybe in bile or something, and then it was probably just because it was suppressed by other stuff, or I don't know, probably was still even in there. So like a lot of things are just missing that should be there. And this ties into the next point, which is the reporting of exotic isomers over common metabolites. And this one is a bit frustrating to me because you see, so when you measure, for instance, a plasma sample and people annotate as sugar, and the only sugar that they've annotated is mannose. Now, that makes no sense because you have very high glucose levels in your plasma. So you can't be reporting mannose without reporting glucose. That doesn't make any sense. You should, like, if there's multiple options for an isomer, it should be the one that makes that you know has the highest concentration, right? Mannose is just some sort of, it's glucose. Okay, so now the cause for this is because a lot of people just use commercial metabolite standard sets. So you have these, I think from Thermo or something, you buy these or you buy these kits, and they have like 600 metabolite standards, and they're a great start. But there's all kinds of things in there that aren't necessarily metabolites, that are just things that some people might be interested in, like paracetamol metabolites, which can be found in plasma. For instance, when somebody has an overdose, it might be interesting to measure them. So they're in these sets. Also, a lot of plant stuff, food stuff, this kind of thing. And again, the artificial sugars. But these in general aren't interesting for you, because if you're looking at, for instance, a cell culture, so different kinds of fibroblasts comparing to each other, you don't care about any of that stuff. And the other thing is that a lot of people use the database from like HMDB, and they don't really curate them, and they just throw them over their M/Z lists, or they curate them a little, and you just, well, this is basically the stuff that I see a lot. I hope it doesn't, it might sound like I'm being very negative about other people doing metabolomics. I hope it doesn't really come across like that. This is just, these are just the things that I've gotten on my desk a lot, and I just really want to warn people about not doing this. And if you are doing this, then maybe this is a chance to, stop doing this. So I want to go through some lists that I got from other groups. And again, this is going to sound a bit like critical, but we're scientists, so we have to be critical of our own work and of others' work. So also, if you've seen things that I've done in this that I'm saying here, and you're like, well, actually, let me know, because I also want to improve. So this is a list that I got from a European core facility. I will say it wasn't a Dutch core facility, just to clear myself. But so what this was a brain set, I think, and they had 96 metabolites in the reported list, and 25 of these are just straight up nonsense. So I have no additional information on exactly what they did, but I just got the list of metabolites, and I just went through these, and 25 of them are nonsense. So just to go through a few of them, so for instance, cellobios, which is like this cell wall thing from, this wall thing from plants, succinonic acid, these kind of things, but also, so this is what I meant earlier. So they reported mannose, but they did not report glucose, fructose 6-phosphate, cellulose 5-phosphate, but not ribose 5-phosphate, which is the much more common version. Itaconic acid, as I said, arbitol, which is again a fake sugar. Malic acid, not endogenous. Maleic acid, not endogenous. Salicylic acid, I think, is an aspirin metabolite. And for instance, G3P. So there's no additional information on what this is, and you have two metabolites that can fit this bill, which is glycerol 3-phosphate or glyceraldehyde 3-phosphate, which are completely different metabolites. And I have no idea which this one is. Like, one is in glycolysis and the other one is in like lipid biosynthesis. And I don't know which one this is. So there's a lot just outright nonsense, and I haven't even gone through the whole list. I haven't even, so I couldn't look at the peaks. I couldn't look at any of that stuff and the in-source fragments, etc. I couldn't check any of this. So just based on the names, I saw that around a quarter of the list was it's just not what it says that it should be. So a quarter is just nonsense, which I think is a lot. So then I got a list from a company. They reported 110 metabolites, and basically the entire list is questionable. So again, a lot of these fake sugars like mannitol, azelaic acid, which is found in grains, so a food thing, and then a lot of isomers with the same retention time. And I put some of them up here. So you see that they have the same M over Z, and they also have the same retention time. Now, this was a case where they thought that they had validated the list by doing MSMS. But again, it's just very common fragments. So for instance, this one, homoserine and threonine, they both have the same retention time, basically the same retention time. Maybe this is a split peak, but probably just the same peak that's integrated, and so the same M over Z. And likely, if you fragment these, you will get the same exact spectrum because they're just extremely similar. So, and again, fake sugars. So it's like fake sugars, and it's the isomers, and they have the same retention time, so just nonsense. And then things like D-ornithine and L-ornithine, which just doesn't really make any sense biologically either. Leucine and norleucine, but then not isoleucine, which is the isomer that you would expect. And just so just in general, things that don't really make any sense. So if you have a list like this, it just doesn't really make any sense. It doesn't help you explain metabolism. And also, if you report this and something is statistically significant, like, what does it mean? It doesn't mean anything. If you're not expecting people to be taking these fake sugars in your data set, like if the people that you're investigating, they don't have this, then why is it in the list? So then finally I have an untargeted metabolomics list, an untargeted approach. And for a lot of these, so I knew what kind of extraction they were doing, and it's an extraction that separates between very polar and apolar. And this is the polar phase metabolomics. So I see a lot of very long-chain fatty acid containing lipids. So for instance here there's a 25-25, so it has two very long-chain fatty acids on it. There is not a chance that is in this extraction. So it just isn't there. I know for a fact. So based on just the chemistry, you can sometimes figure out that something isn't there. The same goes for some retention times. If you're using, especially if you're using a C18 thing, but also if you're using a very polar helic column, if you find a very apolar compound somewhere halfway in your run, that's not correct. Like I know that is incorrect without knowing anything else about your set. So I see a lot of drug metabolites, which we saw before. I also see a lot of picomolar range hormones, and this isn't the whole list, by the way. It was like hundreds of metabolites. So picomolar range hormones that I know for a fact that you aren't detecting. Then plant metabolites again, and some lovely ones, by the way. So we have avocadine, which just, I mean, is that from avocado? Yes, it is. And then you have coriandrone. So we basically have a whole... just a lovely palette of food. And also you need the artificial sugars, of course, to give it that little bit extra. So we have some lovely food related items in the data set. And just to round it out, we have some toxins. So there's somewhere here, acetyl toxin. I'm not particularly familiar with this one, but I doubt people just walking around and being normal people will have this sort of thing in them. And this is another thing, like if you were to take some of these lists seriously, the people that were involved in these studies, like the actual subjects of the studies, they would be dead. So, it doesn't make any sense. So now that we've gotten this out of the way, so we have poor chromatography and everything's just a bit crappy. We have all of this in-source fragmentation. We have annotations that are just not good at all. And so the obvious next step is to throw it in an automated pathway analysis thing, and when you look, for instance, on metabolomics pages on Reddit or something similar, and people are asking like, what should I be doing with my metabolomics data? A lot of them say like, I'll put it in MetaboAnalyst, and you can get like the pathways, and this is very interesting. Like I said, we don't have great data sets in general, so things that are going in aren't great to begin with. But I would just like to show you what happens if your data set is curated very heavily. So this is made with a data set that I curated by hand. So I know that the metabolites that went in there are actually pretty good. And I asked it, so it was about an MMS19 patient, which is it has to do with iron sulfur clusters. And so I added the polar metabolomics list into MetaboAnalyst, and I was like, okay, so these are the metabolites, and you can even indicate like which ones are changed. And so I did this, and then it spat out these pathways. So I've highlighted in red what is just plain wrong. So either the it's actively contradicted by the data set what this says, or it just isn't in there, like there's no proof for it. Orange is when I thought like, oh, I mean kind of, but not really, or it was so vague of a pathway that I was like, oh yeah, okay, maybe. And green is what it got correctly. Now, I've noticed something when a program. So if you, as a human, make any of these suggestions, then people are like, oh, okay, well, why did you suggest this? But if a program makes this, then people start to really rationalize like what came out of it. So, for instance, if I say phytanic acid peroxisomal oxidation is changed, you might start thinking like, whoa, well, what is my thing? Well, it was iron sulfur clusters. Is there maybe a link with peroxisome? So you really start to think about how can I make sense of this? And people do this a lot in literature. But actually, you should really check what's underneath it. Like, why is it concluding this? So for the phytanic acid, for instance, the reason that it's concluding this is because of ADP, ATP, and NADH. The most generic cofactors in basically all of biology. So NADH is used just in tons of reactions basically to power the reactions. So it donates some electrons. And ADP and ATP, it's basically the same thing. They are used in reactions, and it's just the most generic stuff ever. So if I had suggested phytanic acid based on this, like, I would be laughed out of the room. Like, that's not a thing that just makes no sense. Other things just aren't in there. So, for instance, there's arginine and proline metabolism. I checked arginine and proline. They just aren't different. Like, neither of them are different. And also, you could say, like, yeah, but maybe, like, in the path. No, there's just nothing in there that warrants this conclusion. There's also like a lack of biological knowledge in these things. So from other people for publications, where I'm a reviewer or where people send me their manuscripts before they want to send them out for publication, I see things coming from plant pathways, etc., just like nonsense. Of course, you can probably filter some of this stuff, but people don't. So there's like a lot of the time it's just straight up nonsense. So one of the things that I saw as well, lactose here. So lactose is apparently, the lactose pathway is apparently up because of UTP and UDP. Actually, they have a really big DPD deficiency in enzyme, and so their whole pyrimidine stuff is deregulated. So indeed, nucleotides and pyrimidines, this is very much correct. But lactose, I think lactose is only made by pregnant women. So very much no. This is a boy of 7 years old. So unless he has like no. But also, like, one of the problems is that so the way that this works is just it takes a metabolite and it just checks if it's in one of the lists of the pathways. So it doesn't understand whether or not that's really central to the pathway or if it's somewhere, surrounding the pathway. So one of the things that I see a lot is you have these metabolites that have a really sort of strong name. So for instance, glutathione. So people know glutathione depletion, this means oxidative stress. So if I find something on glutathione, I can then say something about oxidative stress. But then in the glutathione pathway, there are things like glutamate and glycine. So if you now have glutamate and glycine being like the levels being changed, then you can get a hit on glutathione metabolism. But glutathione itself isn't changed, and if you have oxidative stress, glutathione itself must change. Like there's no, like, if you have severe oxidative stress, it must change. Like there's no way that it isn't. And what I see a lot is that people then try to publish glutathione metabolism was changed, therefore oxidative stress. But in reality, their data set disproves this very idea. And I think this is just really bad. It just makes for very bad literature, and it just really confuses the stories. Also, you're trying to explain your data, which might be on some new disease that you're researching, because you're probably researching something new. And this is all stuff that other people have already done for other things. So, for instance, in this data set of the MMS19, he missed the pathway thing missed a massive sulfur dysfunction. So every single metabolite that was that had a sulfur in it was just completely dysregulated, and it just didn't see this because that's not one of the pathways that it knows. And also something else was missing, so creatine metabolism was completely dysregulated, and it just wasn't in here. So again, you have false positives and false negatives, just as you have in the lists of metabolites where I see certain things that shouldn't be there, but also I don't see things that should be there. So this isn't great. So summarize. So we have analytical tools that don't really work here. We have poor peak shapes, which makes integrations difficult, and we have pathways that largely overlap. So, for instance, aspartate and glutamate, like there be like glutamate, alpha-ketoglutarate, and you're in the TCA cycle. Aspartate as well, like oxaloacetate things. Like you can, like anything is connected to everything. So, like they all overlap. So if you do like a pathway analysis, yeah, there's just gonna be stuff is just gonna be popping up. So it's very difficult to to do this in an automated way because it's just all pretty And really summarized, incorrectly annotated data sets without internal standards are now analyzed using flawed pathway analysis tools. So that's basically what you see from most of the publications. So just a lot of it is just nonsense. So if we go back to the beginning and all of the issues that we saw in the reproducibility crisis, I hope this kind of sheds a light on it already. I hope you kind of get an idea now on how bad this is and why it's so bad. So I want to end on a positive note. So I don't just want to say like, oh, all of this is bad and you shouldn't be doing this. I also want to say like how I think you should be doing it and how we've been doing it and how it's worked for us. So it starts with a very solid study design. So if you know what you're going to be studying and you have some hypotheses going into it, like I know Untarget is hypothesis generating, but still you know things about your data set. So you might know that your patients are very tired, or you might know that they break out in hives, or you might know you know things about your patients. You know things already before you even start. So a solid study design, and also asking yourself the question like, do I even really expect changes within known metabolites? Like, do I even expect TCA cycle to be different in what I am going to be studying? Like, does metabolomics even make sense to do? Then I think it's important to curate peaks by hand using expert knowledge. So you should know your individual peaks. I know it takes a lot of time, but I think it saves you a lot of time at the end with all of the fancy corrections that you have to be doing and all of them. I see a lot that they have all these horrible data sets and then they start doing super fancy statistics just to sort of get something out of it. I just don't think that's the way. I think you curate the peaks by hand. But there is an annoying paradox there, and this is that doing a better job of curating them means you have fewer metabolites. So if I during this process I threw out fumarate because I saw, oh yeah, it's just completely swallowed by this malate insert fragment, then I won't have fumarate in the data set, but I did a good job. So I will have to report to another researcher fewer peaks. And I think this feels really bad. And I also think, I'll come back to this point a bit later, but what happens a lot is that people then do crappy untargeted metabolomics and then they do get the peak and then they're like, oh, see, this is a lot better. Like, why didn't you get me this peak? So that's a very hard sell, and it just makes it very, it also, I've noticed that showing these peaks and showing what you've done start to finish, like this is how we do it, is not really done a lot. And I think it's because of all of this insecurity on what we're all doing, because we all kind of feel that it's probably not great. So I would always manually check pathways for aberrations. So first you curate the peaks yourself. You should have faith in the peaks that you have, using standards, using internal standards. And then once you have this, you should manually check the pathways. You can use MetaboAnalyst so to get an idea maybe which pathways to look for, but you should always go back and check by hand, like, okay, wait, why is it saying this? What metabolites are different? And what does this tell me? And is this really the pathway that it's saying? Like, oh, maybe actually it's like this. Maybe this is the most logical connection. You should play around with the metabolites a bit, in the sense that, like I said, aspartate can be in all kinds of different pathways. Like you can tie it to the urea cycle, the TCA cycle. There's a few more. But it's also just an amino acid, and so you should check your data set and see like what makes the most sense. Like in what part of the story does this metabolite make the most sense? Is it a change in TCA cycle, or is it just that, well, I'm looking at a cancer data set and all of the amino acids are different, which can then also be linked to the TCA cycle. But just, in general, just try to find like the logic in them. And then, and this is very important, perform corroborating experiments. You don't have to get everything from your metabolomics. So I see people sort of desperately doing all kinds of statistics, like I have to be certain about this metabolite. I mean, not really. Like you can even work with those very, very bad data sets. So if you have that, like don't throw them out. You can still use them. Just realize how bad they are, and then in the end, so even if 90% of the data is bad, if 10% is good, and that 10% results in a clue as to where you should be searching, great. I mean, you've done a successful experiment. The only thing is, you should corroborate this. So expect it to be bad, and then you do a Western blot, do a functional assay, do a fluxomics, so add some labeled comment. If you think like, oh, well, maybe the TCA cycle is different, feed them some labeled sugar, like your cells or whatever, and check if really, the labeling goes down in the TCA cycle, etc. And I've been ranting against the MSMS, but if you have a specific peak that you think is very interesting from an untargeted, from an untargeted set, then just buy the standard, do some MSMS, spike the sample, do some, just verify that you actually did this, because sometimes you'll spike a sample and then you suddenly see like, oh wait, the peak is split, and it's actually not really the thing that I thought I was looking at, because again, horrible chromatography. So then I want to make a point about untargeted versus targeted metabolomics, because untargeted, like I hear people ask for untargeted metabolomics, can I get untargeted metabolomics? But I think that's typically very misguided, and I think untargeted metabolomics, like truly untargeted metabolomics, is not very interesting for most projects. So semi-targeted metabolomics is a way to study known pathways. So for instance, what does glycolysis do after exercise? So I've checked all of the glycolysis metabolites using standards. I know the retention times, I know the peak shapes, I know what they look like in different sets. And so if you want to know, okay, so I have these people and they did some exercise or whatever, did their glycolysis pathway change? Sure, then I can just check those metabolites and I can tell you, did their glycolysis change? I don't need new metabolites. I don't need to do untargeted things, because you just want to know, like, do they have mitochondrial dysfunction? Do they have this, that, blah, blah, blah. Use targeted or semi-targeted metabolomics. And with semi-targeted, I just mean, so like we do MS1, you have the standards, you know the retention times, you curate the peaks by hand. Semi-targeted metabolomics, you get like 100 targets, 150 targets maybe, something like this. Then a very important point, which I kind of made earlier, but untargeted metabolomics is not a way to get more peaks. So I really want to stress this. So if you look at your targeted metabolomics data set and somebody painstakingly curated this, and if you do this for a living, I know your pain. So if you didn't just want more peaks because you didn't see anything significant, this really isn't the way to do it, because I throw out peaks by hand, and then you run an untargeted list, and then you get those peaks back, but they are nonsense. That's why I threw them out. But now you have noise that's integrated, so you just added more crap to your data set for no reason at all. So this isn't a way to get more peaks. It will just create more trash. And if a target is good in untargeted metabolomics, why wouldn't I just add it to the targeted list? So if it was part of one of the important pathways that are just, universal, why wouldn't I just add it? So you're not going to get any metabolites that are in, well-known pathways that I know I can measure well, because otherwise it would just be in my targeted list. So what is untargeted metabolomics for? So untargeted metabolomics, to me, is a way to look for novel metabolites. So, for instance, we do a lot of research into metabolic defects, and these can lead to, well, I said new pathways, but they can lead to new metabolites. Well, sort of new pathways, I guess. So, for instance, we have a disease called PKU, something. You might have noticed I'm clearly more of a chemist than a biologist. So we have a disease called PKU, so it has something to do with phenylalanine, and these people kind of break it down. And they have so much phenylalanine that their cells are like, where do I put this stuff? So they start putting it in the attic and the basement and everywhere. So you find these lipids that have just phenylalanine attached. Or you'll have phenylalanine, which is just, two of them together. because there's no way for them to get rid of it in a normal way. So they're like, okay, well, I'll just try to, not die from all of this phenylalanine that I now have floating around everywhere. And so they add it to everything. So they make metabolites that none of us are making because we can just expel excess phenylalanine. So for a metabolic defect like this, it makes sense to do untargeted metabolomics because we haven't seen these metabolites before, so we need to check peaks that aren't there normally, and you will see like massive increases in some peaks, and then, well, that's kind of interesting. Something like glutathione conjugates also works. So glutathione binds things that are toxic. So if you, for instance, work in a factory and there's all kinds of toxic smokes, or if you smoke, or if you just, in general, you're just exposed to things that are very bad for you, there's a good chance, maybe drugs, there's a good chance that glutathione conjugates form with this toxic thing attached to it. Now, combined, this will have a mass that's not known yet. Like that's a novel metabolite. So it's just a glutathione conjugate, but it's a novel thing. So you could run an untargeted metabolomics thing looking for glutathione fragments, and then try to ID the conjugates, which I think is very very interesting. Again, you have basically novel metabolites. This also works for drug metabolites after a treatment, for instance. So you might break these things down, and you might be looking for the metabolites of this drug. But the chances are just very small that these approaches are going to lead to, just new metabolites in normal people. That I'm really not sure if metabolomics is the way to go there. It might be, and I'm not trying to, if that's what you're doing and you're really focused on this and you have like really specific applications, I'm sure this can work. But just in general, my point is that in general, this is not really what most people who are doing metabolomics are doing. Most people just have like some patients who have some disease or cancer or something like this. And again, cancer could also be producing novel metabolites, but then you really need to understand why you expect this and what you expect. Otherwise, you're in this part, like you're just adding random trash. Okay, so there are some things that you can do for automation, so I want to quickly highlight these. So you can do some blank subtraction, so you have slightly better chromatography. We tend not to do them. I tend to just check it by hand, like the blanks, and check if the peaks that I'm integrating are also in the blank. Now, interesting point, in-source fragments, by definition, have the exact same retention time, because it happens after the LC. Same retention time, same peak shape. So you could check for everything that has exactly the same retention time, and then in-source fragments correlate to the parent ions. So if you have these stacks of masses at certain retention times, then you could calculate like which should be probably the parent ion. You can also check, for instance, based on these mass losses. So they lose logical things. So H2O, a phosphate group, disulfide bridges tend to break. So cysteine tends to break into cysteine. These kinds of things, and so you could check this sort of thing. And then, like I said, they correlate to the parent ion. So any mass that's at exactly the same retention time that correlates throughout all of your samples, likely an in-source fragment. So you could automate this a bit. It wouldn't help you with the horrible retention, like the horrible chromatography, but it would help you somewhat. So this might be worth looking into. Maybe some people are already doing it, and I just don't know about it. But that's an idea. And I want to end on a positive note. So this is the end. Thanks for staying if you listened to the entire ramble. In this paper on the reproducibility crisis in cancer serum biomarkers, it said that an analysis of the type of metabolites reported by 2 or more studies showed that amino acids were commonly dysregulated across all cancer types. So there was stuff that's just sort of that was universal and that did make sense. And amino acids are typically quite easy to annotate. Like not all of them, like glycine tends to be annoying, and there's a few with some isomers and stuff, but they tend to be quite okay to annotate. And so I think that in general, these studies probably, even though only like 24% of them got glutamine for some reason, but I think in general, if you annotate things correctly, they work. Like metabolomics works. We've used metabolomics in, like I said, hundreds of projects, and I've seen our results validated time and time again by corroborating experiments, things like Western blots, etc. And I think it really works if you do it properly and if you really understand why you're choosing a certain version of it. And like I said, you curate these peaks, you go through them. And also, so if you get data from a metabolomics core facility and you pay good money for this, because this is expensive stuff, then you have a right to ask them, like, how did you do this? Like, how did you check for these things? What does this peak look like? Like, if you have a good hit that you think, like, this might really be interesting, ask them for the extracted ion chromatograms, or look them up yourself if you can. But like, if you have a good hit, just take this time, like, go through this. And if you're running something like a metaboanalyst thing, check, like, you can check, you get this readout where you can check which metabolites were used for a certain suggestion. Okay, so I think this was it. I hope I didn't like completely break everyone's enthusiasm about this omics field. I personally really like metabolomics, but I think it requires a lot of knowledge about the metabolites themselves. So really, you should get acquainted with what metabolites do, and so they're not just these random names, but you really understand, like, oh, wait a minute, glucose 6-phosphate, that's here, and this connects to this then, and the pentose phosphate connects like this to the glycolysis, and these kinds of things. I might make a video on this later. And I really think that, okay, so if you're just a PhD researcher who just got some of this data, maybe that's difficult because it really takes a long time. But if you are working in a core facility doing metabolomics, I would really, really encourage you to know the metabolites, like to know what they are, to know where they fit into the stories, because it really helps with the interpretations. So that's it. Thanks for listening, and maybe see you in the next video.